It's truly a great honor for me to be given the chance to talk here at this meeting, and it's also a tremendous joy to meet so many good old friends again. So thank you very much, Dr. Ansari and Tom, for putting together this meeting. Now, as you already heard before, I was in the lab around the time when Marsha Rosner and Takao Sekia, my chairman, were there, and I was a member of what Marsha referred to yesterday as the chain gang. So when I left MIT in October 1976, the lab had just finished the synthesis and of the suppressor tRNA gene and the demonstration of its biological activity in vivo by genetic methods. At that time, I took away not only the very good, useful lessons for a scientific career already mentioned by several colleagues, I also took away a lab member as my wife, Rebecca. So for us, Gobind has always been not only an admired scientific mentor and a wonderful friend, but also a matchmaker, which might not have been planned that way. But just to say, Gobind, thank you very much. Since then, I spent most of my professional efforts on questions of DNA damage and repair. But as the French say, you always return to your first love. And so I more recently started a project that involves gene synthesis again. And in Gobind's presence, I would not dare to say as a sideline, but it's certainly being done with a very small number of people. And I thought this meeting would be the appropriate occasion to talk about this project for the first time in public. So this is going to be about combinatorial gene synthesis used to find new enzymatic catalytic functions that had not been realized before. Now, if we think about how natural enzymes improve their functions or acquire new functions, we would normally envision the occasional point mutation on which selection acts, and then this would result in an ordered trajectory through sequence space. This might not be the complete picture, but it's good enough as a working model for successful biotechnological applications in the sense of what evolutionary protein engineering is doing. Now there is an alternative to that, and this would be that you try to produce as large a library, a random library of structural genes, and then start selecting or screening from that library any interesting any individual member with interesting new function. The problem here is, in contrast to what we just heard, when you're doing this with structural genes, you cannot possibly think of dealing with 10 to the 15th molecular species, because you have to squeeze all your library for the bottleneck of transformation, and then you're down to 10 to the 6th to the 8th or so. And you would like to be very careful about how to populate your library. You don't want to waste library space on candidates that will never make it anyway, because you're touching the folding or you put in amino acids at other places that would not be helpful with creating a new function. So there comes in the challenge of how to tailor a library such that the chances to find something interesting are maximized. And that's what I would like to start with. To introduce the system we're using, and this is the tin barrel or beta alpha 8 barrel proteins, and the first argument in favor of this is purely statistical. More than 20% of all structurally known enzymes fall into this class of folding. There's an 8-fold repetition of this strand, loop, helix strand, and the whole thing folds up in such a way that the eight beta strands form a parallel beta sheet that curves back upon itself in space, and the helices curl down on the outside. Now we used a particular candidate from this family as a starting structural scaffold, and this is the HIS-F from Thermatoga maritima, or in short T-HIS-F. And this is the reaction that is catalyzed. It's a rather demanding reaction, but it's of no further consequence for the rest of my talk, so I skip over this briefly. This is the same kind of look you saw before. From the top view means that it's this side of the barrel where all the beta strands have their C-terminal tips. From the side, it looks like this, and this is the channel in which the substrate lies, and it's a general property of all gene barrels that the catalytic side is on this upper side, and the rest is can basically contributes to furnishing the necessary folding stability to keep everything in place up here where catalysis is supposed to occur. So we can separate the stability part of the protein and a catalytic part of the protein, conceptually at least. And we would like to place all our mutations in the catalytic part and keep this part of the molecule constant. So there comes the planning. This is again a view from the top, and we have 4 layers: blue, green, and there's 3 layers here: blue, green, and orange. The top going down, and then this is the bottom of the pocket. And we identified about 26 amino acids around this binding site, where the amino acids project their side chains towards the substrate that could be imagined lying here. So these are the sites we would like to specifically address in randomization to come up with a library, and we would like to keep the rest constant. Now, well, how do you do this? And the key to this is to use trinucleotide coupling blocks. We use them of this nature. For this particular project, there were commercially, we purchased them from Plant Research, and here you see sort of a picture of good old triester chemistry and Marv-Corazza's phosphoramidite chemistry down here, and dimethoxytrial group up here, everything as usual, and they couple. It's lower than normal phosphoramidites, but good enough to bring about near complete coupling. And by doing so, now you can, whenever the gene synthesis reaches one of the positions you want to randomize, you can apply a mixture of these trinucleotide blocks, and you would need 20 of them, one for each amino acid, and then you can locally randomize. So this idea has been around for a number of years. It was first spelled out by David Shortle, and Johns Hopkins University took a patent on that. It has for some reason not been used a lot, but this method furnishes full control over the site, the degree, and the nature of the randomization. And what I mean by this is the following. For instance, if we add 28% of other trinucleotides to 72% of the residing codons at a given position, we would have 7.3 mutations or exchanges on average. If we build a library with 26 randomized positions and 6.7 exchanges on average, with a library of 24. And they would be distributed in a binomial fashion like this around the mean. So these are the Newton classes, no mutation, 1, 2, 3, and so on. This is the mean would be, the average would be 6.7, and this is the distribution we would expect. Now, for the trinucleotide mixture we do not apply an equimolar mixture of all the others, but we orient ourselves with the occurrence of amino acids on surface of proteins or in catalytic sites, and then the black bars are the molar percentages we use as sort of a compromise between surface and catalytic. We do not want to have a large propensity of having highly lipophilic amino acid side chains in the binding pocket, because these are the kind of residues that would typically reside in the folding core. So now comes the actual synthesis. We have 14 oligonucleotides or polynucleotides to be synthesized chemically, and the black ones contain no randomized sites, and the red ones between 2 and 5 of these sites to be randomized. The number 5 was had a red version, so to speak, when we make the library with 26 exchanged residues, and a black version of it with 24. These are now, at this point, I skip a number of technical intricacies which are important but rather tedious to talk about, and just give you some of the results. These are the 14 oligonucleotides already in double-stranded form. We ligate them together in groups to give 4 intermediate fragments, B1 to B4, and finally we ligate those to the final gene, 786 base pairs. The gene does a bit of flanking sequence for cloning, and then this goes into a vector, and we now have hopefully a library. So now comes the analysis, and the first concern, of course, again, is library size. I mean, we have clones, a certain number of clones controlled by transformation. As I said, typically 10 to the 6 to 10 to the 8 per individual transformation experiment. Now, a subset of these are complete reading frames, which we find out by DNA sequencing. Another subset of these, and this is an important subset, are actually stable proteins. Can we really expect that all our mutants, even if they contain multiple mutations, are stable in terms of folding? And this is the really relevant library size. And we will analyze this by proxy, and I will tell you in a minute what kind of proxy we use to do this. And finally, there would be carriers of function within this relevant library size. And as for now, we use genetic complementation to find them. And this subset, of course, can be larger or smaller, depending on how demanding the function actually is you are looking for. So, first of all, so far we sequenced 239 candidates randomly picked of library 24 and another set for 26, which I will talk about. This is the comparison of the expected and the observed distribution of mutations, and you see this is pretty much similar. And the average exchange propensity found was 27%, expected 28%, according to the mixtures we started with. So for the sequences, 239 clones is 186,420 nucleotides. Wrong nucleotides turned out to be very rare. I mean, all of this was sequenced. Wrong nucleotides are very rare. Deletions are the problem. deletion of 1, 2, or 3 nucleotides. These at these numbers here at mutated sites, and these at the non-randomized positions. And you see delta 1, 29 at the randomized positions means most likely that we're dealing with contamination with dinucleotides from making the trinucleotides. And all in all, it means that we have 40% breeding frames and 60% intact frames in this library, which at first sight seems to be rather bad. But if you do with just a tiny bit of mathematics, you can convince yourself that you have to, the chemistry has to be very good in order to save 60% of reading frames through this entire procedure. So now the next point, stable proteins. We do that in such a way that we take randomly picked candidates and express the protein, and then just look how much protein is made, and is it stable against protease. It is known that generally speaking, unstable proteins either are not do not show up in the soluble fraction because they unfold, precipitate, or that they are rapidly degraded. And this was really a pleasant surprise that we have a very large proportion of proteins that are just as good as the wild type control. This is the wild type control, induced, uninduced. This is a tetracycline promoter, and this little band doesn't disturb us very much because this is also present in wild type. Here we have a case where we have protease sensitivity, and the 3 red ones are controls, clones with reading frames, and you see no protein at all. So we conclude that we have something like more than 80% of the subset of clones with no frame shifts are really properly folded. Now, the last thing is now, and the number of mutations in this set here, in this dark blue, is between 2 and 11. So there is no preference of say, only smaller numbers of exchanges being allowed for the provision of enough conformation or stability of the protein. Now comes the last point I want to make, complementation. We have just started with this, and the first thing, of course, is do we find clones again that will complement a deletion mutant of the original his F. Thermotoga maritima, his F complements the other his F in E. coli, which is important to know. And we started with the thermostable enzyme in order to have a little bit of allowance to lose structural stability before the whole thing falls apart. So now we do a transformation experiment played out on minimal medium with antibiotics. So we select for the presence of the plasmid, and we also select for the activity of going through the histidine anabolism. And so we have we pull out a number of mutants, of candidates, and I can tell you we could actually get hundreds of them if you wanted. This is the negative control, the empty vector, red the positive control, the wild type gene in the vector, and here you see growth curves of different candidates spelled out here. And this, I think, is the last slide already. We sequence 7 of them, and we see of 26 residues, 21 are invariant, at least within this small set of 7 candidates. 1 is completely wild type, and the others have between 1 and 3 mutations in 5 of the sites. So the H84 seems to be quite lenient in accepting mutations. Serine 225 can be mutated easily to threonine, which is not much of a surprise, and then we find these 3 residues being represented by 1 mutation each. So this is lesson number 1, of course, in this study. This is the flea wall, so to speak, and but that's how far as we got last week. So let me summarize. So we use trinucleotide mixtures for controlled randomization. We apply a hierarchical assembly scheme. We can safely store subfragment libraries, which is an important issue, because if you have a highly diverse library, they are known to lose diversity upon storage. We store the subfragment libraries at low diversity and newly assemble the library for each complementation experiment, which is rather easy to do. The bottleneck is still transformation. 10 to the 6th to 10 to the 8th per experiment is not a lot for looking for something you would expect to be rare, some other function more demanding than his F. This is not the end of the story. We're working on loosening the bottleneck, but that would be another talk. Thank you very much.